Okay, we should start. It's a great pleasure to have Ola Jensen here, and Ola has this background in electrical engineering, and he is also an experimentalist working on human cognition, and I think this will be also a very exciting topic. Please.
Yeah. Yeah, thank you very much for the opportunity to come here and present to you. So I hope also through the course of the lectures here we can have some interesting discussions. So don't hold yourself back if you have questions or want to discuss. So my background was originally in electrical engineering, but then I switched to neuroscience. And then I went on to do cognitive neuroimaging. So what I'm trying to do today is sort of to link what we have been doing in cognitive neuroscience to some developments in AI. So just to set the stage, so personally I'm very inspired by this field of neuro AI, right? So I mean, there's no doubt that AI is undergoing a revolution right now, right? So we're all aware about what you can do with ChatGPT and all these tools that is being brought forward by AI. I should say this is a very recent thing, right? So you might have heard about the AI winter. So like decades ago, AI had a lot of promises that were not really fulfilled. People got disappointed, but now things are really coming back with vengeance, right? And I think everybody are very impressed what AI can do with very simple algorithms. So as a cognitive neuroscientist, I think we can be inspired by these developments in AI. So one thing I can put just ChatGPT as one example, right? But this is an algorithm based on transformers, which is trained on a lot of language material. But what it does is it's being trained to sort of predict the next upcoming word, right? And by this very simple principles of trying to do predictions and then adjust these predictions if they are not fully correct, then these algorithms can do a lot more than one might have anticipated initially, right? So the idea here is, that by looking into some of these advancements in AI, maybe we can also inspire new thinking in cognitive neuroscience about how the human brain works. Yet the other perspective is that the human brain is still so much more efficient than AI, right? So if you consider a baby, this baby learns a lot within 1 year, and the training material that baby is exposed to is a lot less than what our AI algorithms are making use of, right? So AI can also learn from the human brain in the sense that the human brain can learn to generalize on a much smaller training data set. So this is to say that I think both AI can benefit from cognitive neuroscience and cognitive neuroscience can benefit from AI. So this is sort of the space we try to operate within. And I will do this by putting forward a concrete research question that we are trying to solve. We have not solved it yet, but we are trying to make progress on that. Oops, that was the wrong video. Yeah. So this research question is inspired by the way humans explore visual images, right? So it's clear to all of you that when we look at an image, our eyes are circling around all the time, right? This is an old eye tracking study demonstrating that the eyes in this case are moving from person to person, and in particular from the head area of this person. So what's going on here? Well, it's supposed to be some sort of accumulation of evidence as we are exploring this image, right? So our brains didn't have to work this way. One possibility is that we sort of look at the full image and then sort of are acquiring information and then analyzing that. But that's not what's going on. We are sampling this image actively, right? So this is different than how our typical AI systems in vision works, right? Of course, there's some breaking down of the image and moving around and so forth, but this systematic sampling and accumulation of evidence seems to be unique to humans, and maybe this is a good way of analyzing visual images. Furthermore, when doing this, there's also some sort of accumulation of information going on, meaning there needs to be some sort of memory in the system, and that's different from how a convolutional neural network works, as we will talk about later. Okay, so this is the problem we want to solve. How does the human brain accumulate information by this sampling? And might that process also inspire algorithms in AI associated with vision? So now to some interesting mysteries when we're doing this sort of sampling. The first one being this issue of repetition suppression. So it's a very simple thing. It has been shown many times that if you show an image and then show the same image just half a second later, there's a suppression of the evoked response of that image, right? So it's called repetition suppression. So how does this repetition suppression work with the accumulation of evidence, right? It's not obvious. However, one principle that might come to the rescue is this notion of sharpening. So the idea is that first time you show the image, you get a response. Next time you show the image, you get a lower response. But it could be that this lower response is associated with a fewer number of neurons coding for the second representation. However, these neurons code for this more precisely, so to speak. So there's fewer neurons partaking in the coding the second time around. But these are the neurons that really partake in the code. And then there might be some other neurons that coded for this image initially, but this coding was less relevant, so to speak, right? So this is the notion of sharpening. Associated with this accumulation of evidence, you also supposedly get a more and more precise neuronal code, right? So this is something we can investigate in the human brain or the animal brain, if you will. And then we can also start to think about neural networks that is doing this kind of sharpening. So,
sorry,
yes.
I'm a little bit confused with this sharpening. Is it that means the activation will be higher or it becomes sparser?
It becomes sparser, right? So fewer neurons partake in the coding the second time around, and that means that the activation actually is lower, albeit is more precise. Yeah?
Thank you.
Yes?
So this might be a dumb question, why do we call it sharpening if the response is lower?
Well,
What's getting sharper exactly?
it's the representation itself. So it's more sparse, as you put it, but it's fewer neurons, and it's only the neurons representing the core representation part.
Oh, so you're kind of whittling it down.
Exactly, yeah. Good. So in order to investigate this, we can start by asking some questions. The first question would be, do some of you have ideas to how we can study sharpening in an experimental setting in humans or animals, if you will? Yeah, so that's a very good question. That's a very good suggestion, right? So we just have a multi-array recordings in some visual area and then repeat the stimuli. Now comes the problem, how can I do that in humans if I only have access to, say, both imaging with functional MRI or MEG or EEG recordings? Yes? Oh, so, yeah, okay, so the question being, how can we study sharpening in humans using EEG and MEG recordings, or functional MRI, if you will? Because there we do not have access to the individual neurons, right? And how would you look for a neuronal code using these many electrodes.
ICA components? Just something.
Yeah, ICA. So you guys are getting there, right? But what we'll be using is multivariate pattern analysis, and I'm going to explain more about that. Okay, so the next question for you to think about for now is, say that we construct a deep neural network, like a convolutional neural network, and we want that to show something like sharpening. How would we go about this?
How visualize the weights of neurons to have a better propagation?
Yeah. So we can look at the neuronal code, so to speak, in those networks, and then repeat the stimuli. What will happen if you show a deep neural network and sort of freeze the training of that, but if you show the stimuli the second time around? It's sort of a rhetorical question, but in the sense that nothing will happen, right? Because there's no memory in these deep neural networks, right? So we have to think about how these deep neural networks works in terms of repetitions and maybe having some sort of memory built in, right? So these two questions are sort of going to be the core of what I'm going to tell you next, right? So since this is the tutorial session, and I understand some of you are theorists and some of you are animal electrophysiologists and experimentalists, then most of you are probably not familiar with MEG, so I'll start telling you a bit more about that and how MEG is relating to electrophysiology in animals. So what you see here is a typical MEG system. So this is a system we have at the Center for Human Brain Health in Birmingham. The idea is that we have a helmet here in which there are sensors, and these sensors record from the human brain. So we're going to open this up in a minute, and I will tell you what's inside this system. But of course, the idea here is that we are recording the ongoing brain activity from human individuals. And what we are doing is that we are recording at the scalp level, right? So we have hundreds of sensors distributed over the head. But what we want to do is to infer down, ideally, what's going on at the individual neuron level, right? Of course, we cannot do that directly, but we can do that indirectly using multivariate pattern analysis, right? But nevertheless, MEG, there's nothing mysterious about it, in the sense that we are convinced that it is the neuronal activity resulting in some currents being produced in the brain that are producing local field potentials, and eventually potentials and fields that can be measured at the scalp level, right? So even though you might be an animal electrophysiologist, MEG is not that different, except that it's a different spatial scale that we are measuring on. So what is going on is the fact that if you have neurons in the brain, we know that many of these neurons are nicely aligned, right? So if you look at the cortical lamina, we have the deep pyramidal cells that have these dendrites going through all the cortical layers. In these dendrites, there would be currents generated. So we now know if currents are being generated, there will also be a magnetic field, and that is what we can measure at the scalp level, and I'll show you some example of that. There will also be a return current from these dendritic currents, and these return currents are going to produce different scalp potentials that we can measure with EEG, right? So first of all, if you're familiar with EEG, it is the same basic electrophysiology generating the EEG signal that also generates the magnetic fields that we can see at the scalp level. So how do we relate these electrical currents in the brain to magnetic fields? Well, this goes back to a discovery by this guy, Hans Ørsted. So he's Danish like me, so in Denmark we are very proud of him. Nevertheless, what he did was to take a compass, and then he had an electrical, then he had a wire that he hooked up to a battery, right? And when doing so, he saw a deflection on that compass, right? So probably no big surprise to you, right? But back then it was the first study linking electrical currents to magnetic fields. More or less, what you now can imagine is that you can have a neuron like this, a nice layer 5 parameter neuron, say, and there's a dendritic current in this neuron. This current might be generated by a synaptic input at the apical dendrites. And then we know from the right-hand rule that if you take your thumb and align that with the dendrite, there would be a magnetic field going around. So in principle, what you can imagine is that you put a compass next to your head, think really hard, and maybe you will see a deflection on that compass, right? So, of course, that's not going to work because the compass is not sensitive enough. But what we can do is that we can have a more sensitive magnetic field detector. And the way we make that is simply by having a loop, a wire loop, a coil, and then you hook that up to a simple ammeter. And then you measure the magnetic field from the dendritic currents, right? So when you talk about MEG, it's nothing else than this. It is this simple. It's simply having a lot of coils distributed over the head, and from these coils we measure very small currents, and these currents are generated by, we measure very small fields, and these fields are generated by currents within the brain. Right? That's MEG, no more, no less. This is the first MEG system made. It was made by this man David Cohen, working at MIT. So this is a shielded room where he did the first recordings. And the way he made the magnetic detector was to have this magnet here, or coils with about 1 million windings. So this was super sensitive, right? So you might have tried this as kids on your physics lessons. Take a nail, take a wire, and then loop around that, then nail, and then you can make an electromagnet. Since you can make an electromagnet, you can also use this system to detect changes in magnetic fields by taking the same setup and then hooking up your little setup to an ammeter, right? And this is exactly what David Cohen did, and he was able to measure the magnetic currents in the brain reflected by these fields recorded by the coil. Nonetheless, this is not very practical because this coil here is huge, right? Yes?
Maybe a technical question. So, like the distribution of the dendrite currents in the brain orientation is different. So I guess the magnitude of the brain also like very different directions. Why not they cancel out each other, or and then why they have some together they have a large magnitude?
Yeah, no, that's an excellent question, right? Because you have the gyre and the solid side, and then you have neurons turning in different directions. So then you think that if you have dipoles generating magnetic fields and they're all differently oriented, they will cancel each other out, right? But now it turns out from a practical point of view, they don't. So it must be because the patches of activation is sufficiently small, not to be spread out over neurons with different directions, right? And we know that deep layer neurons, like for instance layer 5 parameter neurons, have dendrites that are aligned perpendicular to the brain surface. Of course, they sort of loop around the soil side, but when you take and add this whole thing up, you actually get quite a sizable signal outside the brain. What you should also remember for MEG, if you have radial dipoles, and that corresponds to dendrites being perpendicular to the brain surface, they would actually not measure a field that we can measure outside the brain. So it's mainly from the walls of the soil side that we see these currents that generate the fields that we measure. But it is a bit surprising that that things don't cancel each other out. But empirically, from an empirical observation, we get very robust signals. Okay, so this setup here is quite impractical. So a typical MEG system are made by these so-called superconducting quantum interference devices, right? So basically what that is simply a coil, as we talked about before, and then it's hooked up to a very sensitive ammeter that is constructed by means of a superconducting device, right? So what you need to know here is that part of the circuit has to be immersed in liquid helium in order to operate, right? So it's based on superconductivity. You don't necessarily need to know the details of that. All what you need to know is that we have a very sensitive ammeter that measure the magnetic fields produced by the brain. However, since this has to be immersed into liquid helium, we need a big thermos bottle to keep the helium cold. So a typical MEG system is big and bulky, right? So we have these systems where you put your head in, as you saw at the picture before. The system is very big and inflexible because you need vacuum insulation to keep the helium in there. Nevertheless, the good news is that it works as a charm, right? So this technology has been around for a few decades, and when you buy an MEG system, you just plug it in and it works. Nevertheless, it is also inflexible, and if you want to record from children or infants, that's a problem, right? So we talked about before that maybe we can learn something about, maybe we can learn something from the infant brain, the baby brain, because babies in the first year learn a lot, right? With not so much stimulus material as, say, an AI system. So if you want to study that, this MEG system is too inflexible. Nevertheless, there's a lot of excitement about a new kind of MEG technology called optically pumped magnetometers. So this works by a very different principle. It's based on rubidium atoms that can be aligned with polarized light. And then if there's a magnetic field from the outside, this alignment of the rubidium atoms would go out of orientation, so to speak, with respect to the polarized light. So you can then measure how much light you need to pump back in order to align these rubidium atoms again. So to make a long story short, what this cell here does is that it can measure magnetic fields from the brain based on an optical method that does not require liquid helium. So that means nowadays we can do MEG with these more flexible helmets, and it works just as well, but we can adapt the helmets to individual children or even infants, right? Yes?
How many magnetometers are in the system?
So as many as you can fit in.
How many?
So I think in Nottingham they have, I could be wrong, but something like 250.
So it's a bit more than like EG system.
Yes, and EEG system you can also do. I mean, you can also do hundreds of electrodes. It's just then you have the trouble of making the connection for each electrode, right?
So it's kind of much easier to like install the system.
That's the thing, yeah. So if you have a 4-month-old baby and you want to do EEG, there's quite a challenge on getting the EEG system working before the baby loses his or her patience, right? In this kind of system, you can just stick in a baby and then start recording. And you ask how many sensors. Now it turns out that these sensors, we can actually measure the magnetic field from 3 orientations, right? So you might actually get 3, pay for 1 when having these OPM systems. So I think it was a question there first.
So what's the main challenge to use this system?
So it's basically engineering. The sensors are there. Now it's just about scaling up the system and get the sensors stable. So it is happening. I think at Peking University, just around the corner, they have a lab that they are developing and applying these sensors as well. So there's several companies doing those. I'll show you here. This is our system in Birmingham, where we have about 80 sensors. And we have a system up running, it works. We also have a collaboration with physics where we can make our own sensors. So you had a question?
And the subjects move or do they stay still?
They can move a bit with the helmet, but you don't want them to bob around too much, obviously, because then you get artifacts. But they have more freedom to move than a conventional MEG system. Yeah? Yes?
Do we attach the magnetometers to each other to be able to record with the odometer signal?
So a conventional MEG system has magnetometers. These OPM systems work by gradiometers. You measure the gradient in the magnetic field. These sensors here, as the ones you can buy off the shelf, are magnetometers. Now it turns out that our team in Birmingham has developed the first gradiometer. So if you look at this guy, then you can find a paper out now that they've been able to make a gradiometer. So a lot is happening. Yes?
Sorry, I missed it. Did you already comment on like, the compromises from the system versus the full MEG system? Where you lose resolution or
So the compromise is that the noise level of the sensors is slightly higher, but the signal magnitude is also higher. So the thing is, with conventional MEG, you have to be maybe a few inches away from the scalp due to the vacuum insulation. The magnetic field drops by R squared. So you lose a lot of signal that you can gain by putting the sensors closer. So you get a 4 to 5 fold stronger evoke response from the auditory cortex using these OPM sensors. Then the noise level floor is also a factor 2 higher. So it's a bit difficult to sort of talk about the signal to noise gain per se, but we do seem to get a more better spatial resolution. This will be relevant for doing multivariate analysis I'm going to talk about next. But this is so new, so it's hard to say anything concrete about the signal to noise gain.
the general idea is that it's actually better than MEG?
Better or just as good, and just as good is even advantageous because the liquid helium is very expensive also to recycle, and furthermore get the flexibility that you can adapt the system to children. You had a question?
I was going to ask, what's the cost of this thing compared to average? What's the cost of this thing compared to average?
Oh, so an MEG system would probably set you back about 2 million euros. So this year is actually not too different in costs because the sensors, they cost about 10,000 euros, and then you can sort of scale up. But you still need a sheeted room, which will cost you about 500,000. Then, so me being a center director, I have to worry about maintenance costs. Then the maintenance cost for conventional MEG is a lot higher. It's more than £100,000 a year for MEG system. For OPM system, it's less. So it's difficult to say, but it is expected to get a lot more inexpensive with time.
So the operational cost is slightly lower than MEG?
Yes, because the whole liquid helium and liquefier and so forth, that's what is costly.
Is there a way to also do it on the scalp like EVO? Or
To put it on a scalp, in principle, you could already do that now, but then you have to lift off the skull and then have measure from the brain, but then people have to be anesthetized.
yeah, like EVO, but like, can you do that with magnetic?
You can do that. The question is, why would you do it? Because if you have open to the brain anyway, then you can put the electrodes on, right? But people have been doing this in animal preparation and slice and stuff, so it is in principle doable. Yes?
I want to ask you a question about the traditional MEG system. Everyone's been a subject for an experiment. I found that traditional MEG is so uncomfortable and so low noise.
Yeah.
Where is there such a low noise?
Oh, so you're probably talking about MRI, right? If it's noisy, where you have the switching gradients from the MRI system. So MEG is completely noiseless, so it's more comfortable than MRI. Okay, so I'm actually not really here to talk about OPMs, but this is just to say that a lot is happening in this field. So while MEG hasn't changed too much in terms of hardware for the last 2 decades. And now finally something new is happening with OPM, so people are quite excited about this. Anyhow, what I'm going to talk about is multivariate pattern analysis. So this is a relatively new approach to looking into human MEG and EEG data. And people have become quite excited about this because this is a way of bringing us closer to the representation of specific information, right? So back in the days when looking at animals, people could look at representations by considering individual neurons, and now you have the multi-electrode recordings. And of course, us working on humans have always been jealous about that. Nevertheless, one way of getting closer to that level of representation is to use multivariate pattern analysis. And I'll first explain how we go about that. The idea is that, of course, if you look at evoked responses from the brain, then you can introduce visual stimuli and then you can look at the response. We now know that here you perceive these circles and squares differently, right? More or less the evoked response from those might look very similar, right? So that is of course a bit problematic if you want to look into representations, in the sense that you could take a monkey and then you could record from say like a color selected area, and then you will be able to tell the red from the blue, for instance, right? Nevertheless, we cannot do that in humans. So if we look at these evoked responses for the blue and the red circles and squares, then we see exactly the same response, right? So that is, of course, a bit disappointing. And for decades, people have been looking at these evoked responses. Then you say, well, look at oscillations. You also know these stimuli induces modulations in oscillations. It's the same, we cannot tell these two stimuli from each other. However, if we are using multivariate pattern analysis, it actually turns out to be possible. So the way to think about that is that we are doing a multi-array recording from the brain, and what you have here on the X and the Y axis could be EEG or MEG channels. It could also be voxels with functional MRI. And what you then see is that the red squares seem to collect down here in the lower left corner, and the blue circles they collect up in the upper right corner, right? So if you can make an intersection between these two clouds of responses, you might be able to tell the representation from each other, right? So this actually works. What we can do is that we can do recordings with EEG, and then we can look at the time course, and then we can ask at what point in time is the brain able to tell the squares from the circles, right? You can also do it with the both responses from MRI, and then you ask where in the brain can you tell these two stimuli from each other, right? Yes?
What is the unit of the axis?
Oh, so that all depends on what you're doing. So if you are doing EEG, it would be microvolts. If you're doing MEG, it would be femtotesla. If you're doing the board signal from functional MRI, it would probably be some sort of a stat score. So it could be anything. So the idea is, as I mentioned before, is that you can do a lot of trials of these, and then you can look at how these distributions of data separate with respect to the stimuli, right? So here we're only looking at 2 channels, but say that we have 300 channels, then of course you have to think about a 300 dimensional space that you are making some sort of surface in that space separating these 2 clouds of data, right? So what one typically does is to train a classifier. So it could be a support vector machine, it could be linear discriminant analysis. There are a lot of those. It could also be logistic regression. and then you train that on a subset of the data and then test it on another subset, right? So of course, what algorithms you use is making some differences, but our experience is that it doesn't actually matter too much, right? The most important thing here is to have a robust paradigm with good data. And as I just mentioned, one does a cross-validation. So in this case, this is just an example of a 10-fold cross-validation. The idea is that you take 90% of the data and you train your classifier in order to find this boundary between the two classes of data, right? So after you have trained your classifier, then you can test it on the last 10% of the data, and then you move on to another 10% of the data and so forth. There's many ways of doing this. It doesn't matter so much. Some people do 5-fold cross-validation, other people do leave one out, right? Nevertheless, this has all been developed and implemented, and it works quite well. Yeah?
I just had a question about terminology.
Yeah.
I don't know, I've noticed that MBPA is mostly used in the human field, and less so in machine learning more generally or in neuroscience. So I guess what is the boundary of that term? Like, what does it designate as opposed to just SVM or LVA, which is applied more broadly across fields? Is it just kind of nature of the data set plus these other methods, or is there something distinct about the method relative?
No, so as a multivariate pattern analysis is sort of the most general term, and it encompasses SVM, LDA, reputational similarity analysis. So it's just to look at the data in a multivariate sense.
So I guess the idea is that you don't do dimensionality reduction prior to passing it to the SVM. So if you were to reduce dimensionality of your data before doing that, you no longer want to call it MBPA.
Yeah, so I mean, so typically you don't want to do too much dimension analysis because it's sort of already taken care of by the algorithm, right? Yeah. But the key point is that if you don't do something multivariate, you cannot tell the different the responses from the different stimuli apart. If you do look at the multivariate data, you can. Yeah. Yes.
What about multi-class classification? How many classes can be distinguished in this method?
Oh, so it depends on the data. So I mean, in principle, you can have 5 or 10 classes, and then there's different approaches for how to do this. You can do, if you have 5 classes, you can do 1 versus before.
Yeah, of course, 5 classes is like the bigger, but like 1,000 classes or even more.
Yeah,
well,
but then,
too many representations in the human brain, and I'm curious about how many can be actually distinguished like with this particular method.
Yes.
Is it bigger than EEG or?
well, so the way to think about it is that typically you probably need sort of 30, 50 trials per category in order to be able to tell things apart, right? So that also sets a boundary for how many classes you can have. So I would say it's probably more practical concern for how long your experiment is that allows you to collect enough data to get sufficient classes.
But actually easier to do it with MG rather than EEG, right? So the classifiers can become more like accurate.
That we would like to there's a discussion on this, but in general I think that's correct. And that's so I will not go into details, but too much detail, but it turns out that EEG signals are much more smeared than MEG signals. So you get a better spatial resolution with MEG and even better with OPM. So we have not proven that case yet, but we would like to at some point be able to demonstrate that OPMs are the most optimal approach to use multivariate pattern analysis. Yeah?
How about the generalization ability of the classifier?
Across participants?
Yeah.
Yeah, so what you cannot do is to train a classifier in one participant and then test it in another participant. There's some sub-cases where it's possible, but in general, you cannot cross-generalize across participants. So your representation for a dog versus a cat is unique to you, and so if I train my data it will train or classify data from your brain. I cannot use it to test my brain and vice versa.
So can you use from trial to trial and just such as you train data for me, and the other day I do another experiment.
Yeah. So it will be stable for you over time. Yes. Yeah.
Just to make sure I understand it, you need a lot of classes like dog, cat, other animals, other categories to make sure you have enough multivariate data to then tell them apart, tell the responses apart.
So I'll get more into this, but just for this support vector machine type of approach, you only need 2 classes, but you can have more. But I will move on to representational similarity analysis, where we can talk about more classes. So I'm afraid of running a bit late. So since you asked many good questions, so I'll just skip this part here and then move straight on to representational similarity analysis, right? So this is sort of meant to be more of a generalization of the multivariate approach. And the idea is that if you have a lot of objects like this, right, there's different ways you can group those. So you might see here there are animals and there are flowers. Maybe they are grouped in a particular way in the brain. It could be that you think about colors. So here you can see there's something yellowish, oranges out to the right, something bluish, something greenish down here, right? Could it be that of course we know that our brains can do that kind of grouping because you all understood what I said when we are grouping according to color, right? So information might be organized according to color. Another possibility is that you group according to category. So here we have animals, we have clothing, flowers or plants and so forth, right? So it could be that the brain group stimuli according to those principles. And this is all something we can test now. And the way people do this is by sort of deriving the representational dissimilarity matrix, right? So what we are doing here is that we are correlating activity associated with these different stimuli. This could be brain activity, it could be neuronal activity, it could be brain activity based on neuronal representations, but it could also be activity from a neural network, or it could be behavioral responses according to how similar people find things. So if you take this, what you should imagine is that we have all the stimuli here lined up, so there might be 100 stimuli, so we line them up according to this matrix here, and then we can cross-correlate whatever measure, right? So this is a dissimilarity matrix. So the more dark, the more similar things is, right? So this first square here represents different animal representations. So you can see they are quite similar. However, animals are different from clothing objects, right? So here you get something more reddish, yellowish. Nevertheless, animals might sort of be more similar to plants because it's something to do with nature, right? So what you can look at this dissimilarity matrix, you can look at the broader category level, right? So that would be these big squares here, but you can also look at the individual item level, right? So you have this complex representation reflecting how similar or not these different items are with respect to a given measure. What one can do is to construct these so-called IDMs according to behavioral data, as I mentioned before. You can do it according to the brain data. It could be intracranial, single unit recordings, but it could also be MEG and MRI data, or it could also be the activations of units in a deep neural network. So I will show you an example done here. So this happened to be children that was shown different kind of stimuli. So these are visual stimuli, toys, bodies, houses, and so forth, right? And then the ongoing EEG was recorded. What then the researchers do was to look at how similar or not the brain activity was when considering two stimuli, right? So it could be that you present a toy or you present a body, and then you look at the similarity or the dissimilarity in terms of the brain activity. So for now, probably the most easy way to think about this is that we look at the correlatedness between the activation at the EEG level, right? So toys of different kind would result in quite similar activation and therefore quite similar representations, correlations in EEG signals at the SCAT level. Yeah?
This is all over the brain, or can you find that it's not in regions?
So for now it's all over the brain, and then we'll get into localization a little later. The key point is that now if you want to look at the category specificity of these RDMs, what you can go back and do is to make a model matrix, if you will, sort of where these dark squares here are 1 and the other squares here are 0. And then you can relate that to your experimental RDM and see how well they correspond. And you can do that time point by time point, and then you can ask at what time in the brain do these representations start to form, right? Yeah?
Because all offline data after it was pre-processed.
Yes. Yeah, so it has to be offline. Yeah, right? So the idea is that you look at how things groups in the brain, and by grouping I mean the correlatedness of, in this case, the EEG data, and then you can ask how well that grouping occurs according to category, and then you can look at the correlation, look at these correlations as a function of time, and then you will know at what time point the brain can sort of form these representations. Yeah?
We would have like similar studies, just different stimuli, because like these categories are kind of, I mean, we already know that these categories are differently represented in our brain.
Yeah.
I mean, we already know that according to ERP studies, we don't need an additional processing to understand that child will respond differently to toy, body, pulse, and face.
So I mean, if you do ERP studies, indeed you can see there's a slightly stronger face response, like insanity compared to houses, right?
Yes. So for example, with bodies, we will have like activation of motor cortex because like we just basically respond to that. Toys are kind of defensive for it.
So one could do that. So it is possible in some cases to tell, say, faces from houses, right?
Yes.
But in general, I think you would be hard pressed to find brain responses that correspond differently to different houses or houses different colors, or even looking at body parts versus faces. They also become hard to tell from each other, right? So the question is not whether the brain can tell these things apart. The question is more how well can we do this categorization so that we can exploit that information later. And the funny thing is that, so I should actually show you the next one here. So I'll just skip ahead. But it turns out that the phase response is at 170 milliseconds, right? In 170. But if you use this RSA approach, you get the representation of specific information somehow earlier. So there, actually, when you use the RSA approach, then you find that the categorization happens at around 120 milliseconds, right? So even though you're right about the evoked responses that tell some apart, using this approach, you actually get a very different time. Anyhow, yes.
Is there any history pattern when you do this, such as something you think is similar, but the representation is quite different, or
Can you say that again?
Is there anything strange happen when you do this kind of analysis, such as something you think is a similar thing, but the representation can be quite different?
Yeah, I don't know exactly how to answer that. Could you give an example?
Just such as the animals in the same kind of in the same class, but
Yeah.
but maybe one animal just different from others.
Yeah, I mean that's possible. So one could like design an experiment to check that, right? How would how would a strange animal maybe be represented different than a common animal? So what could look at that? Anyhow, these experiments have been done a lot, right? So here's another example where people have been looking at bodies, faces, animate, inanimate, and so forth, right? So the beauty of this is that you can start to group these different object types differently, and then you can ask questions to how the brain might be representing those in different ways, right? So again, this is an MEG study with these different stimuli has been shown. There's a classifier being trained, and that allows us to look at the representational dissimilarity matrix that you have here, where you see the grouping according to animals, non-human, and human stuff, and so forth, right? And then you can look at the brain activity as a function of time. And the key point here is that now we can go in and say, you can isolate. In this case, this is hard to show, but this here pertains to human body parts. This is to human faces. So what you simply can do is that you can sort of make a model matrix where all is here is ones and the rest is zero, and then you can correlate that model matrix with the value M measured from the MEG data as a function of time. And what you then can do is to look at how fast these different representations activate as a function of time, right? And the key point here is that at about 150 milliseconds, you get a peak for being able to tell these representations apart, but the build-up happens even before, right? Yes?
So I understand here that the point here is that the difference between the lines, not like the absolute scale on the y-axis. But I'm just curious, I see someone has tried just other classifiers, particularly like more recent ones, picture-based mechanisms or CNNs. I'm just curious how much those lines would or would not move up the y-axis when you apply nonlinear approaches.
Yeah, so I don't know exactly the answer to that, but we have been playing around with this using just different kind of classifier like support with machines versus ND and so forth. And it doesn't matter too much, so it might change things a few percent, but you get more or less the same pattern out of this.
The LDA and SVM are more similar, my understanding, than opposed to SVM versus like some kind of over-parameterized network. So get some sense for what would happen if you apply these more recent models to it. Does it bump it up more, or do you still have to see only a small change in percentage?
So I don't know, but my feeling would be that it might improve a bit, and that's always good, but it's not going to change things dramatically. Right? So the key point is that this here is fairly robust, but there will be sensitivity changes. Good. Moving on. So here comes some of the things that's quite powerful about these methods, and this also pertains to the localization as was asked about before. Because you can do this approach with MEG, but you can also do the same approach with function MRI. So since the time course is quite poor for function MRI, what you then can do is to have a searchlight that you move around in the brain and then look at the port signal. And then from the port signals, you can also do your representation of dissimilarity matrix. Right? So what you now can do is that you can show a face and then ask when can a face representation be reflected in MEG signals, and then ask, when you have this face representation, when is then that the strongest correlated with RDM derived from the probe signals, right? So that means you can get you can fuse data from MEG and functional MRI and get the best of both worlds, right? So for MEG, you would know when in time a face can be choked from the data, and from the probe signals you can tell where in the brain the face is represented. Right? So you can do a lot with these methods to fuse things across different recording modalities. Does that address your question? Yeah. I mean, so there's also the possibility of doing a searchlight approach with the MEG data in source space, but we don't have time to get into that. Okay, so moving on. Then you might also start to ask, well, could it be that some of these representations we see in the brain are akin to the representations being formed in deep neural networks, right? So no doubt that convolutional neural networks have been very useful for classifying images, and they are based on these structures that at least some people would say are quite similar to the structures we find in the mental stream of the human brain. So could it be that some of these representations that emerges in the human visual cortex and the monkey visual cortex are akin to the representations that emerges in these deep neural networks, right? And by similarity, we mean that the sort of grouping of the different objects together as reflected by the RDM would be similar in the human brain and the network, right? And that is something we can do now, right? So as I mentioned before, we can fuse data between MEG and functional MRI, but we can also sort of fuse data between a deep neural network and MEG. And this is what I'm going to show you here. You're probably all familiar with convolutional neural networks, right? So a lot of what sparked this was the so-called AlexNet. So that was a type of network having these convolutional layers initially, and then you have higher and higher level representations, and then the classification in different categories, different items here at the output layers, right? And this network was trained by a standard back propagation algorithm, and then trained on a huge data set of images, right? And it turned out that this was quite powerful, and has later been different evolutions of this. Now it turns out that this network, the AlexNet, actually have quite some similarities to the human brain, in the sense that the early layers have these convolutional properties, and then you have the sort of more overarching all-to-all layers further down the network, right? People have argued, well, since these networks are so similar to the human or the animal brain, maybe they also represent information in a similar way, right? And this is what other people have been trying to test. So the idea is that as before, we have a huge number of stimuli, then we present that to the human brain, we construct the RDM, and we can look at those as a function of time. Now we can also train a deep neural network to exactly classify these stimuli as you ask the humans to do, so to speak. And what you then can do is to calculate the RDMs for the individual layers in the deep neural network, and then you can correlate those together, right? And the conclusion here is that indeed there is quite some good correspondence between what the deep neural network does and what the human data do, right? And in particular, if you look at the early layers of the deep neural network, they correspond to the representations early on in the human brain, and the deep layers in the deep neural network doing the high level classification. It could be humans versus animals versus foods, right? They do correspond to what's going on at later time points, right?
There's another question.
Yeah?
How do you test the dependence
benefits of these results on the architecture?
Yeah, so the architecture is just imposed by the machine learning, right? So it's not like people are trying to say there's so many neurons in V4 and then we need therefore so many units in a given layer in a deep neural network, right? So this is just a sort of a very rough approach where one is not trying to mimic the architecture beyond having convolutional layers and then having the higher level representations.
If you add more layers, is change these results?
You can play around, but the general picture doesn't change too much. So we're later going to discuss the issue of time, because you can say like maybe it takes about 5 to 15 milliseconds for activity to propagate from one layer to the next in the human brain. If you only have 120 milliseconds to play with, as shown by the MEG data, you can maybe not have more than 6-7 layers in your deep neural network, right? So one will start to play around with these constraints, but that is not really done. The key point is that people have been trying many different deep neural network architectures, and they all sort of form representations that are somehow similar to what we see represented in the human brain. Yeah?
This question is kind of a detail, so if you outline, you want to move on, but I'm curious like how you actually go between the neural network and the brain, because you can't do like a pixel-wise correlation between the heat maps because like the permuted between no correspondence or the dimensionality is different. So when you go from like your middle plots to your right plots, how do you align things or sort things appropriately to get that similarity?
So if you have 100 stimuli, your representation of dissimilarity matrix is 100.
Oh, I see. So you're aligned by stimuli.
Yes.
I got it.
Yeah, and all what you do, and it is as simple as this, you simply take your REM for your MEG data 100 by 100 and your REM for layer 8 of your deep neural network, and then correlate those two together. And this similarity is that correlation. So I mean, if you can do correlations, you can do representational similarity analysis, and you can do this analysis. Yes.
Just from curiosity, like people ever try to like select some item that you never seen from a kangaroo? For example, somebody never seen panda, and then but they already seen other animals, and like for CNN, we are sure because these pandas share some feature close to other animals, so maybe the representation are similar, but what about the frequency?
Yeah.
There,
It's an interesting question. So I don't know whether that has been done, but it could be an interesting study to perform. Yeah. So I think I have to speed up a little bit, but nevertheless, here's the point again, that you can look at the different layers and how that corresponds to the human brain activity. And again, this has been done a lot, and the general conclusion is that what's going on early in the visual system corresponds to the early layers of the deep neural network, and what's going on in the deeper layers corresponds to what's going on at later time points in the human brain. And this is, again, relating this to functional MRI data. But I think I'm going to move on to a study we have been doing here. So as I mentioned initially, what we're interested in is this sort of priming going on in the human brain. And this is a study conducted by Sienna Vin in my group. And what we wanted to do was to go back to this issue on accumulation of evidence, right? So you're circadian around in an image, and your eye lands on different parts of that image, and in that process we are supposedly building up more and more information. What we want to get at is to go towards a more realistic architecture and dynamics of deep neural networks that can do this accumulation of evidence. We're not there yet, but this is what we're working towards. And we talked about the issue of sharpening. So how is this sharpening going on in the deep neural networks, right? Since sharpening, the notion is that the second time around you show the stimuli, you get a more crisp representation coded by fewer neurons, but we supposedly do this more precisely. What we are thinking about how deep neural networks could be changed to do this process. So the study we have been doing is on priming, and what we do is so-called parafoveal priming, right? So in real life, what is happening is that your eyes are moving around between different objects. I'll go here to the one-back prime. So this corresponds to, so now just fixate on this part here where you keep your focus on the fixation cross here, right? And then there's two stimuli to the left and right. So typically you might move your eye to one of them, it could be the car, right? But saccades are creating problems when we are doing MEG and EEG data. So instead of having people move their eyes, what we do is that we now present a stimuli in the center of the screen, right? So this here actually sort of simulates a saccade. Instead of there being a car out there that we then saccade to, we show a car out here and then we present the car formally as if the eye had moved, right? So the key point is we want to sort of simulate the stimulation in the paraphobia associated with an eye movement. And to make it more challenging, we also do a two-back prime. So here you can see the shirt being primed out here in the paraphobia, then there's intervening stimuli, and then the shirt comes back in the phobia after that, right? So what we have been doing is to present these data to human participants, about 30 participants, hundreds of stimuli of different categories, and we are trying to do as few repeats as possible of these stimuli. And then we have recorded the MEG data, and then performed the analysis we talked about before. So the first point I want to show to you is that we get this zero back, we get this repetition effect, repetition suppression as we talked about before. So the repetition suppression is that if you set the car out here, the second time you should have a smaller response. And this is indeed what you see here. That is the one back prime, right? So this is about 100 milliseconds out. If you don't, zero back is no priming. If you do the priming, you get a significant reduction in the both response and equation, right? And the sort of conundrum here is that how can it be that supposedly when you are priming the stimuli, then when you see the stimuli again, you should be able to perceive it better, but the brain is actually responding with a slower response the second time around you see that stimuli, right? So that's the conundrum we're starting from. So what we then did, of course, was to do this classification according to the MEPA approach using a simple support vector machine. So it could be that we have categories like fruits and animals, how that can tell them apart, right? In our study, we had 5 classes that we wanted to tell apart, so we did 1 versus the 4 others. And I think we did a 10-fold cross-validation. And here are the results. So what you should imagine is that on the x-axis we have time, on the y-axis we have the accuracy of classification. And if you just look at these curves sort of more broadly, so this is when the last stimuli is coming, right? The phobia stimuli, following the priming in the paraphobia. What you should appreciate here is about 150 milliseconds in, we have a fairly strong response in the classification, right? So lo and behold, we can classify these objects very well, right? But no big news here, this has been done in many other studies. The key point being that if we look at the one back prime as compared to the zero back prime, we get a stronger response after about 150 milliseconds, right? So this means that if you think about repetition suppression, the second time you show a stimuli, the brain responds with a weaker response, However, if we use the multivariate pattern analysis, we see a stronger classification from the brain, right? So this is consistent with this notion of sharpening, where supposedly fewer neurons are partaking in the second response, but they are doing a more precise job at representing that stimuli. Right? Yes?
Is it true that like after 200 milliseconds, the neuropriming is actually better?
Yes, so the question is what is going on out here, right?
Yeah, and especially because it looks like it's better than.
Yeah. So I mean, one can start to speculate what this might be about. But what you should keep in mind is that we saccade 3 to 4 times per second, right? So after 200, 250 milliseconds is all over and I move on to the next. So I'm not saying it's not interesting what's going on out here, but in a real life situation, we have already moved our eyes to the next stimuli.
So in this time period, what is the accuracy from the 200 to 300?
The accuracy?
Yeah.
So this is measured as the area under the curve. So I mean it's about 0.56, right? So it's not whopping, but it's very robust across participants.
But since the participant has moved his eyes away from in this time period.
So in this case, in real life, people would have moved eye away. In this study, they keep distinction. Does that answer?
Yeah.
Yes.
How large is the error bar?
Yeah, it's a good question. I don't remember, but we have been doing statistical tests and found this is robust. Right? So, but not, again, it's not, I mean, it's significant, but again, it's not a super strong finding. And that probably goes back to the notion that we do MEG is very lousy data, right? And 0.56 of a chance level of 0.5 is not that impressive, but it's good enough to demonstrate reliable effects. But if you want to do a single trial classification, right? So just to show a fruit or an animal, and you want to classify that on a single trial basis, if you do that, you will do quite poor. Yes.
How do we compute similarity in this particular case? You say we do this matrix.
Okay. So this here is a support vector machine where we compare one class to 4 other classes, and I'll go on to the RSA analysis next.
Okay.
Yes.
I'm not sure if this is possible, but since I think you're basing this, coming back to the original hypothesis that this could be due to like a sharpening effect or something like that, can you look at like the features that are important from the decoding from the SVM and kind of see if it's very distributed or if there are only like a few features that it's paying attention to?
Yeah, so hold on, so I think we're getting closer to that next. Yeah, so basically what we now have been doing is to construct some deep neural networks that can at least make an attempt at explaining some of these effects, right? So what we have been using is the standard AlexNet. So it was trained on the initial image set used for the AlexNet, but what we then did was to also then expose it to these new stimuli. And we also put an output layer on that allowed it to classify these stimuli in 5 classes, right? So we pre-trained the network and then updated the training with the new stimuli. So now we have these different layers, and for these different layers, as for the MEG data, we can calculate the representational dissimilarity matrix. So this is some key chains on the network, right? So I don't think we need to dwell into this, but there's nothing mysterious here. It's just sort of bread and butter stuff when doing convolution neural networks. Also for the energy data, nothing too mysterious here. When we are having the stimuli introduced, we had 500 stimuli, so we have an idea of the 500 by 500. Yes?
So it's, for instance, correlation of all the sensors, right? But when we will see the matrix, it's like one measure. I mean, do we estimate the average across all the sensors?
So you show the ice cream, then you show the dog.
Yes.
To get that part of the representation of this matrix. And then you simply calculate the correlation over sensors for that stimulus pair.
Yeah, and I have a vector, like with the size of number of channels.
Yeah, 300.
Yeah.
306.
But in the matrix, there's only one value.
Yes, so it's 306 correlated for the 306 values for the dog correlated with 306 values for the ice cream. Yeah.
Last picture.
Yeah. Okay, so as I mentioned before, what we now do is that we look at the dissimilarity matrix for the brain activity and for the individual layers. And what we then can do, as we also saw before, is to look at the individual layers and how they correspond to what's going on as a function of time, right? And what you should appreciate is that the low-level features are being represented in the early layers of the network, and the more higher-level features that the different categories are represented in the deeper layers of the network. Was that somebody asked a question on this? Yeah.
This is human data, right?
This is human data. Yeah. Right. So basically, what shown here is what other people have shown is that you get higher and higher level representations as you go down the vector scheme, supposedly as the signal propagate, right? So no big surprises here. The novelty to this is that now we can go in and look at these correlations with the network as doing no priming, one priming, or two priming back, right? And this is again relating this to the different parts of the network. So, of course, it's difficult to just eyeball these and tell the differences apart. But what we have here is the details, and I'll now zoom in on first the early layers. And what you should appreciate here is that what we get in the early layers is that we have the stimuli itself. Not being primed, you get the strongest response, but if you prime the stimuli, there's also a lower response in these early layers of the network, right? So this is back to the repetition suppression. So what we would like to argue from this, based on the deep neural network architecture, is repetition suppression is occurring in early layers of the deep neural network. So now if we go to some of the deeper layers of this network, what you should appreciate is that when you compare the one-back priming to the zero-back priming, is that you get an increase reflected at around 180 milliseconds, or from 150 up to a few hundred milliseconds, for the one-back priming being stronger as compared for the one-back priming as compared to the low priming condition of blue line. So that means that this sharpening we talked about before supposedly is going on in the deeper layers of the network. Yeah.
On the prior slide, we have data to control cells that is not occurring like even further upstream, like in the retina.
It's a good question. So we don't have those data. But it is a possibility, right? I mean, you could imagine neurotinol cells achieving a bit, this is a cat priming a cat, right? So this one what I said. So if you have cat, you get repetition suppression. If you have dog, cat, you don't to the same extent.
But importantly, different cats with different pixel arrangements.
Yeah, so that we didn't try. So that would be an interesting next generation of this experiment to go in and ask, could it be that stimuli of the same category is going to prime each other and do repetition suppression and or sharpening?
So these data are currently identical images.
Yep. So that indeed would be very interesting to do that in the future, to ask whether the priming is also at a more abstract level, but we don't know. Okay, so to make a long story short, for the last 5 minutes, the conclusion from our study here, that we have been able to link deep neural networks to the MEG brain activity in the context of repetitions. What we find is that there's repetition suppression in the early data, supposedly also early visual regions, and in the deeper layers we have the sharpening, right? This is how far we got. But what we want to do in the future is to go in and look at the dynamics of this, right? So one issue with these networks is that these deep neural networks, they do not have memory, right? So they do not remember what you saw one step back, and we want to build that in. And we have some ideas for doing this. So the first idea is that in the feed-forward connections, we are sort of boosting the synapses being activated, right? So if you have a complex stimuli being presented, and it's used through the network, the output layers, what you then could imagine is that one pathway being carved through the network doing this is sort of being, that pathway is sort of being carved out stronger, such that when you see this item the second time around, you will get a strengthening in those, you will have a strengthening in these feed-forward connections that also boosts the sharpening. Another possibility is that we have sort of all-to-all connections with each network, right? Within each layer. So it's almost, you can think that you already learned about attractive networks, right? So you can think about one attractor being formed for the first presentation, such that this attractor is going to be sort of re-evoked by the second time the stimuli is presented, right? So we have two specific mechanisms that can account for this type of priming and sharpening. We have not done that yet, but you can see where it goes. We hope to implement this in the network and then go back and ask the data which of these two mechanisms are most plausible. So I think we are right on time. I will end here by acknowledging my research group. Several of these people have been involved in this work, and thank you very much for your attention. Yes?
It could be that I missed it, but how does this sharpening depend on similarity of the stimuli between themselves and the probability for it?
Yeah, so it's a bit pertaining also to the question to whether different categories can prime, right? There's also the probability of occurrence. The sharpening notion has also been elaborated on in the context of predictive coding. So people have been doing, for instance, functional MRI studies, in which they have also found evidence for sharpening if you can predict what's coming next. Right? So if you predict a dog is coming up, you can actually get a smaller response to that dog in terms of the evoked response, but you get an increase in the classification if you were doing that kind of study. Does that address your question?
Talk to you later.
Yeah. Yes.
She's first.
Okay.
Okay. Do you focus like on like simple neural network architectures on purpose? Why not like studying like LSTM or transformers that can like store context and choose?
Yeah, no, I mean, so I talked about our networks don't have any sort of memory, so why not do LSTM or something like that? And the reason being is that we want to keep it simple because we want to be able to map onto the human brain. And I don't know how to map an LSTM onto the architecture of the visual system. But some might know how to do this. But that being said, it is a very interesting discussion because some people have also tried now to take the transformer trained on a lot of language material and then look at some of the layers in that transformer and then map it onto the human brain activity as recorded by functional MRI. And that works very well. And they say, oh, let's just ignore all about the architecture of that model and then ask, are there sort of similar representations forming these models that maps well onto the brain? And in terms of the transformer, that is the case. So it could be that one direction we could take here is to actually entertain other structures and then be a bit agnostic to whether they sort of map onto the human brain architecture or not, and say this is more about the sort of computational principle, not so much about the details of the architecture.
Yeah, so I'm asking about related precision suppression. So you see this suppression in one back experiment. So you expect also the suppression in the two back.
Yeah, so we did have that built in, but I didn't have time to go into the details. So the answer is that we get less of suppression in the two back, and we get less of suppression, less suppression and less sharpening in the two back.
Can you go back one slide priming fact? This one.
Yeah.
So if you look at this, I expect there is some suppression effect in the two back, right? But if you look at the response, the two back response is less than
Yeah. Yeah.
And wondering why that happens.
Yeah, so I don't have any good explanation. So we had hoped that the two back also would be higher than the zero back.
Yeah,
But that is not the case, unfortunately. Any other questions? Okay.
All right, let's take a break.